Glad you guys are here. There's currants of focaccia over there. There's also extra fruit from one of Everle's. That's a lot. Please drink all the coffee. Yeah, so welcome. We're going to hear from Yesuf and Carrie and Russ today. The latter two are online on economic research updates on winter oil seed and soybean cropping systems. And we're happy to have them. As always, I'm going to start by acknowledging Purple Green's funders, the Environment and Natural Resources Trust Fund, Minnesota Department of Agriculture, Clean Water Legacy Amendment, Builders Vision for Philanthropies, and the McKnight Foundation. Thank you for making this work possible. Reminder to folks, we're recording, and people here, we're recording this meeting. It will be online.
Just a reminder that we're hosting a book launch for The Living Roots, The Promise of Perennial Foods. We're going to have Colin Curitan, Forever Green's Director of Commercialization, Beth Dooley, Reginaldo Haslett-Marqueen, and Gwendell Westerman as panelists. And Liz Carlisle and Audrey Strike-Krug will be facilitating that conversation. There's going to be a reception afterwards. It's a free event, so come join us. You can scan the QR code to register.
I don't have a lot to share. I just wanted to just mention again that we will be testifying in the House Agriculture Committee hearing on Monday afternoon. It's just an update. There's not a bill on the table for funding or anything like that. We just want to keep the committee up to date on what we're up to, share the good progress, and share how their investment of public dollars is paying off. So that will be coupled with the broader hearing focused on AGREET, which we now, Forever Green now gets funding through AGREET, including for me, for Colin's position, and for 4 of our breeders. But it's a pretty small chunk of the overall AGREET allocation to the college, which supports dozens of faculty members and some staff members. So we had the opportunity to tag onto that, which is great, and hoping that goes well.
Hey, Mitch, just a quick note there. Apparently the House is doing ticketed hearings this year. Pretty unlikely that the tickets will run out, and I'm not entirely sure what the process is, but if folks are planning to attend, maybe just bake in a couple extra minutes to get your tickets sorted, whatever that winds up looking like.
That's very good to know. Thank you, Pete.
Good afternoon, everyone, and thank you for coming. My name is Yesuf Mohammed, and I am Oilseeds research agronomist. I would like to thank Doctors Mitch Hunter and Russ Gesch, my current and previous supervisors, co-authoring this presentation. So today I will be giving winter oilseed agronomy research that me and my collaborators are doing. Just to give you a brief overview, winter oilseeds are winter camelina and pennygrass. They are very small, tiny seeds compared to corn and soybean, but they are planted in fall, and they provide the functionality of the traditional cover crops, such as protecting soil from erosion, reducing nutrient leaching, as well as providing food for insects. Besides that, they can be harvested for seed, and this seed can be processed for biofuel and bioproducts. The good thing about this crop is that they mainly grow during the off-season on bare field, so they help to minimize the land competition for food, fuel, and feed production. In addition, they help to increase resource use efficiency, such as solar radiation, water, soil water, and land. Because these crops are new to the landscape, we are trying to develop agronomic practices so that they can be incorporated into the existing cropping system. Some of the activities include that we started variety evaluation, which include both camelina and winter camelina and pennygrass variety evaluation. And we initiated 2 separate experiments for these 2 species, and we wanted to know which species or variety are performing better and where. So for the winter camelina variety evaluation, we have about 12 entries, and they are sourced from University of Minnesota Camelina Breeding, Vision Bioenergy, New Farm, and we also use Jewel, Bison, and Dakota Fertilizer as a check. These varieties were planted at 7 locations 5 in Minnesota, as you can see in the map, in this map, and 2 locations in the neighboring state at Brookings and North Dakota at Carrington. The collaborators include doctors Carrie Eberle at Morris, as well as Dr. Russ, same location in Minnesota, and Dr. Shannon Robert at Brookings, both from UCDRS. We also have another collaborator at Carrington, North Dakota State University, Dr. Christine Simon. The variety evaluation was planted at planting data ranged between mid of September all the way to the first week of October. The same thing is for pennygrass, except that we have 10 entries for pennygrass variety evaluation. In this variety evaluation, we included only the yellow or golden seeded varieties. The seeds were sourced from University of Minnesota pennygrass breeding program, as well as David Marks lines. The location and the planting data are the same, and the management are the same with the winter camelina, and probably this will give us the opportunity to compare the 2 species, at least to have some idea which species will perform and which location or site. As you can see in the bottom, for our collaborators, we package the seed per plot and ship the seed to their destination. I wanted to mention 2 important points here. This is a photo shared by Dr. Shannon from Brookings, South Dakota, where they were planting this winter oilseed using no-till system in wet stubble. So 2 important points here. If we succeed at this site, number 1, planting this winter oilseed using a no-till system will help to minimize that input energy compared to planting them on till field as wasica. Number 2, if we plant them under this no-till, under wet stubble, the wet stubble may be able to catch or trap snow, so that might help to increase winter survival of this crop because the snow will act as a blanket. This is a picture taken on November at Brookings. You can see a very good germination of winter camelina, very good plant stand that was taken on November, mid of November. And also the next picture is taken from sample from pennygrass field. So the variety trials are doing very well at both locations based on this photo. Another important activity that we recently initiated is to see the row configuration and the relay soybean date. As you know, relaying is one of the strategies to incorporate this winter oilseed crops into existing crop using a skip row system, which we call it as business as usual, where when we plant the winter oilseed, we skip 1 row, then in the spring, we'll come and plant or relay soybean into it. But we noticed and recorded that using this relay system, business as usual, there will a yield gap from the relay soybean compared to monocrop soybean. We need to narrow that yield gap as much as possible so that farmers can adopt the system. In addition, we also noticed that when we plant relay soybean into standing winter camelina, because winter camelina will be harvested a little earlier. So when the soybean has the same plant height with the camelina, it complicated the winter camelina harvesting. So we are proposing and initiating already established the experiment. If we able to give more space for that relayed soybean, in other words, if we able to widen the row, the soybean instead of itolating, it will probably grow shorter a little bit, produce more branch, and that may help us to produce more pods and also minimize the yield gap. And if it stays a little shorter, that will also minimize the winter camelina harvest complication. So in this experiment that we already established, we'll be evaluating 2 skip row versus 1 skip row in addition to soybean relaying date. So we'll be looking at soybean ethylation, soil moisture dynamics, photosynthesis, active radiation under and below the canopy of camelina, and as well as seed and seed quality of both camelina and related soybean. This project is funded by FGI, hopefully to get the money very soon.
Hey, there's a
Yep.
question on that.
Yep.
Is your seeding rate of the camelina the same, like pounds per acre for the 2 skips versus the 1 skip?
So the rows are plugged. That means less seed is going down. So it is adjusted to that area.
So it's a lower seeding rate.
Yeah, lower seeding rate.
Yeah, lower population.
Yeah, lower population.
Okay.
It's like 6 inch or 7.5 inch or something.
7.5. Yeah. So this is to skip. As you can see, there was supposed to be 2 rows of camelina in these rows. So we skip them. So we are going to come and plant the relay soybean in this row compared to the business as usual. This is a controlled winter camelina that we planted
Usual, but would
with one.
piping area be like 15 inches business as usual, and now you're gonna be back like 30 inches.
Yeah.
You skip rows?
I think it is 7.5 inch row spacing, the winter camelina.
3. Yep, 2 rows, so it'll be 20. 20, okay. That's
22.5.
Thank you. Yeah, that's what I was getting at. The space between the camelina rows for the soybeans is 22.5. Yeah.
Yeah, it's a little wider. Yeah. So very soon we'll be coming and relate or plant the relay soybean on these skip rows. Next activity that we have is seed rate evaluation for domesticated pennycress lines. As you all know, the previous seed rate was based on the wild type, which has a greater seed dormancy compared to recently developed lines. So we wanted to evaluate the new lines because these new lines are very good trade compared to the wild type, including better germination and less seed dormancy. So we wanted to evaluate this seed rate on seed yield, oil content, oil yield, and as well as green cover. So the seed rate ranged from 1 to 9 pounds per acre, and we use the TT2 lines, and we planted this experiment at Morris as well as Rosemont in 2024 fall, and we had the first year result from 2025, and both sites were planted around mid of September to end of September, and harvested around the first week of July using plot combine. So as you can see in this result, on the left side of the graph is showing on the x-axis seed rate, on the y-axis seed yield of pennycress, or Morris with blue solid lines, for Rosemont with orange dashed lines. As you can see for the blue solid lines, seed yield increase with increase of seed rate at Morris, and it reached to the maximum when we apply 7 pounds per hectare. But for Rosemont, seed yield remains almost flat for all seed rate. So this is really very important to see from this photo taken on the mid of May. As you can see, that photo, which was taken at Rosemont, all the plots looks like the same. You don't see any significant difference in terms of plant stand. So we visually observed that for low seed rate, pennycress produce a lot of tillers, and those tillers try to grow very fast and catch up and become more productive producing seed. So probably that is why we didn't record a yield increase when we increase seed rate for Rosemont site. When it comes to the oil content, we didn't see any impact of seed rate at both location and stay on average around 31%.
Yeah, it's pretty wild that 1 pound per acre still yields the 50 pounds. So a lot of what the
Yeah, that's it. Yeah, that's
So
very.
maybe it's fine that you increase the seeding rate for your double skip? Cause like it'll still. Yeah.
Yeah, I think, that's a good point. I think what I noticed between camelina and pancreas, that skip row for the relay system is for camelina. What I noticed from the two crop is that pancreas produce more tillers, but camelina tended to produce more branches. That is something I noticed. So keeping the seed rate for pancreas low might be compensated by more tillers, especially if there is enough moisture and early warming of the soil. Those tillers grow very fast and become productive and produce seed as a mother plant for pancreas.
So are you saying that maybe it's not the same for Camelina then?
That's what I try to.
Is there a seeding, do you have a seeding rate study plan for Camelina?
No,
Okay. Yeah,
it's just from my personal observation.
sure, okay.
Yeah, I don't see more branch in the panicles compared to camelina, even in the greenhouse.
But this is kind of important for seed increases, right? You could plant very low populations and still have decent yields.
Yes.
But the
Yes.
reality is into a production situation,
Yeah.
price of seed is gonna be, you would assume it's gonna be low, right? In most cases?
Yeah.
Who cares if you plant $9 of seed?
I think that's very important point, and I will support that by my next slide.
So Rich has a question.
Yeah.
How do these results for the domesticated pennycress compare to the results for the black seeded pennycress?
This is the first year result, and this experiment is going to be repeated. Is already repeated this year. So we'll have the harvest data from this year. We'll combine the two sides to your data, and then we'll compare for the previous result, and we can conclude something. This is just 1 year result, and it's very difficult to conclude from this 1 year result. And probably next year, this time, we can compare the previous study with this one, and we can say we can put a conclusive remark from two sides to your data compared to the previous study, I think. So, when it comes to green cover, as you can see from the left side of this slide on the graph, where the seed rate on the x-axis, the green cover is the y-axis. So green cover is a kind of proxy for soil cover. So one of the most important characteristics of this oil seed is to provide soil cover to minimize soil erosion. So as you can see, increasing the seed rate significantly increase green cover, which is really very important to cover the soil both in fall and spring. It is really important to consider the soil aspect, because covering the soil not only minimize soil erosion, but also producing more plants means they will take that residual nutrient from the soil and help to recycle that nutrient instead of that nutrient leached into the water body or goes through runoff. So, as it has been said, finding the balance to go to a little bit higher seed rate might be a good idea, because we can provide that soil cover even if we don't get significant yield increase at low rate. That's really very important point to make. Another point that we noticed here is that we measured NDVI, which is a proxy for the greenness of the plant, close to harvesting June 24 and June 30. It might be difficult to see the trend, but the lower seed rate tend to have a little bit higher NDVI compared to the higher seed rate. If you consider only this the orange graph. So the lower seed rate tend to have higher NDVI compared to the higher seed rate. What does it mean is that by keeping the plant green, if we use low seed rate, by keeping the plant green, allowing them to grow more, we delay the harvesting of pennycress, which means we are delaying soybean double cropping. So this is not really a good quality. So in other words, we have to increase the seed rate. So when the plant compete among each other, they ripe very fast, and we can cut them early, so we can double crop soybean, so we can avoid that yield drag from the double crop soybean compared to the monocrop.
And the green cover, was that canopio?
Yeah, canopy. Another activity that we had is evaluating pennycress harvesting method that we superimposed in A2 last year. So we assume that if we able to remove or cut or harvest pennycress early, we can double crop soybean early. So we evaluated desiccant called G45, swathing, direct combining. We compared this harvesting method, and swathing desiccation was done in June 13. And depending on the weather, when you swath or desiccate, the harvesting time depends on the weather following those 3 days, 4 days. But if the weather allows, this process of harvesting through desiccation and swathing will give us 6 to 8 days advantage of seeding double crop soybean compared to the direct combining, because the direct combining was harvested June 24. But we didn't see any significant yield difference between for pennycress among the three harvesting methods. This is a collaboration research activity between the Department of Entomology and our project with Dr. Robert Koch. This is to evaluate the effect of winter camelina and pennycress on Japanese beetle aphids and stinky bug. So we established winter camelina pennycress using skip row at Wasika, Rosemont, and St. Paul. And soybean will be relayed on the skip row very soon. And the entomology people will come and see the effect of this oilseed crop on insect population in terms of number and the damage on the soybean compared to the monocrop soybean. So this project is funded by the Minnesota Invasive Cereal Plants and Pest Center. This is another small activity that we did in the greenhouse. The bison winter camelina that we had was stored for more than 10 years. So in order to increase its viability as well as to increase it, to use it in the greenhouse, we raised the seedling in the greenhouse, transplant them in the pot, and collected some seeds that we used for the variety trials. Another activity we did last year was pennycress seed increase in A2. We have this almost 3 acre in, for low glucosinolate as well as high standard glucosinolate content. We observed that when you reduce the glucosinolate content, the plant tended to delay maturity compared to the standard. We harvested and calculated the yield, and we saw that the low glucosinolate had a little bit less yield compared to the standard glucosinolate, and the standard glucosinolate has a little bit more oil content compared to the low glucosinolate content. Because this was my first time to run this big plot with big machines, and there was a lot of learning experience in terms of how to derive this much amount of seed, how to clean this seed, because we have to ship the seed at the end to Garkin. So a lot of people participated in these activities, and it was a lot of frustration as well. But the facility in MCIA helped us to clean the seed, and you can see here a very super clean pennycress from strash and pots. Finally, we delivered, shipped about 500 pounds of pennycress with low glucosinolate and about 900 pounds with the standard glucosinolate to the Cargill facility in Colorado. So it's a big achievement.
Yesuf, quick question from Carrie. When did you desiccate the pennycress, and did it allow for an earlier harvest date?
Desiccation and the swathing was in June 30, and the direct harvest was on June 24.
How much yield increase would you get from soybeans by planting them 6 to 8 days earlier?
Well, that is a problem because this was not planned to plant soybean after harvesting, because this experiment was superimposed on that A2. So the other plot was not harvesting. So actually we went there, the swathing, we didn't have the swathing machine. We used hand just to mimic the swathing inside the big plot. But from my visual observation, sourcing and desiccated plot in the big plot could have been threshed in 4 days after that, because this desiccant is very, very heavy and can dry out very fast. And swathing, as you know, depends on the weather. If it is dry, it can dry in 2-3 days. Be ready for. But if it is rainy, then it can elongate.
Try to think through.
But we didn't double crop, or you're just
If a producer is thinking about this situation.
Just say, can you speak up?
Desiccating and swathing, it's 2 extra passes into the field.
Yes.
And is 10 days more soybean production worth it to pay for those labor costs?
Yeah.
What are we gonna get for?
Yeah, I think just before you answer, I think it depends. In my experience, 2 days matter. If we plant early and then the next day it rains, then that rain soaks the soybean and facilitates germination of the soybean, and it can make a big difference. But as you said, it has been this kind of study should have been well designed so that double cropping soybean will follow immediately after swathing and desiccating. Sorry.
Half to a bushel per day.
Yeah,
That's pretty big. It's a pretty big drop.
Yeah.
Yeah, it's big. So it'd probably be worth it. Do you remember the desiccant? Was it like a sodium chloride?
Yeah, sodium chloride. Yeah, 45%.
I put a shout out for hyaluronic acid as a desiccant. I
Okay.
read that's a good one. It just seems to, well, from what I read, it just does a better job. I think. Yeah, faster. Yeah. And it's not a salt either.
Yeah.
MID.
That's good to include next time.
It's a fatty acid, so I don't exactly know off the top of my head, but it shouldn't be too expensive. Yeah.
Thinking about the environmental impact story of the winter oilseeds, what are the pros and cons of desiccant?
Yeah.
Is there a negative image to using it like there is in food crops?
Yeah, I think that's a really good point. And sodium chloride, the desiccant that we used here, might increase the soil pH a little bit higher. That might not be convenient for some of the soil microorganisms, but this happens once in a while, and it can be leached very easily with rain. Yeah, I think. Yeah.
Yeah,
But,
I mean, I can see increasing the yield of soybeans is going to make the system as a whole more viable if you have more acres of it, which is a great outcome.
Yeah.
So just thinking about what are the trade-offs if you're using a desiccant, is there an important trade-off that isn't something to think about?
yeah, I think that's a very good point, and I think that is something we have to look probably after harvesting, take soil sample and see its effect on the soil microbial diversity and number. Maybe that's something we need to look.
Research has also already been done just on desiccating without swathing. Is that why you did the both? Like, it seems like overkill.
No.
Desiccate and swath and then combine.
No. Desiccant is you desiccate it and then you come back and direct combine it. Swathing is another.
That was a separate treatment.
Treatment. Yeah, sorry. Yeah. So there are 3 treatments: desiccation, swathing, and direct combine.
Oh, yeah.
Yeah, it's separate. Yeah.
I have a couple of things I need to add from online. Anna Kate said those extra passes would increase CI score, but that might be balanced by higher yield. Mitch also wanted to add an important piece of context is that desiccating and swathing are not options in a relay cropping system. It's still useful to develop this information for a double cropping system, which may be viable further south in our region, or if you are able to greatly shorten the maturity rate.
Yeah, I think I agree with both comments.
Unless you use glyphosate.
Yeah.
I don't know if glyphosate works. Yeah.
Matt, what's the product the desiccant that you mentioned?
Oh, hyaluronic acid.
We do have one final question online from Matt Levitt. Similar harvest yield results for camelina by the different methods.
For camelina?
Yeah.
I don't know.
Similar. You don't know?
I don't know.
I do a lot of desiccation stuff and so. So did you see any kind of more synchrony in terms of the seed dry down, or did the desiccation actually improve any kind of seed quality things, or do you think it was just a time maybe, and they're probably confounded, right? So it moved it earlier, but is there any addition, do you think there's other additional benefits? Did it maybe take out some green seed that you would or high moisture seed you would normally see?
Yeah, I think I didn't, because this pennygrass is very tiny seed, to see individual seed quality is very difficult. But the desiccant might help to dry down those green weeds, otherwise that will contaminate the seed. So it is a good advantage to include things at the same time, because some of the summer weeds try to grow faster in summer and catch up with the pennygrass. When it is time to harvest the pennygrass, the weeds are still green. When you desiccate, those weeds will dry and avoid the problem with moisture, seed contamination, things like that. I think so. That's what I have. Just to mention last slide, I would like to thank our funding source, Cargill Forever Green Initiative, Minnesota Intensive Terrestrial Plant-Based Center, helping the summer part of the research activities. I also like to thank our collaborators, Dr. Carrie, Eberle, and Russ Gesch at Morris, Minnesota, and Dr. Shannon Osborne at Brookings Lab, as well as Dr. Christine at Carrington, North Dakota State University. I also like to thank Vision Bioenergy and New Farm, allowing us to use their material in the variety evaluation. I also like to thank University of Minnesota, Camelina, Pennygrass, and wheat breeding teams for their generous support and sharing of resources. Otherwise, I could have done nothing without their help. MCA allowing us to use their cleaning facility, especially to deliver that seed, clean seed to Cargill in Colorado. Dr. This helps us, giving us driving the hefty tractor when everybody was busy, and he volunteered to help me planting the variety trail in Semport. And also he gave us his experience to get some maintenance on this hefty tractor. As you can see the seat, we usually to bring pillow to sit on the hefty tractor, but he helps us to replace the seat. So that was really very helpful.
So I'm going to start and talk a little bit about a project we've been doing looking at alternative crops to relay with winter camelina, specifically our work with chickpea, sunflower, and safflower. So these first pictures here, we've got chickpea relayed with the winter camelina. The chickpeas on 30-inch rows looks very similar to what we see with soybean. In the middle is our sunflower. This is also relayed on 30-inch rows. Both of those use the skip row planting for the winter camelina. You can see that sunflower is a little bit taller than maybe what we see with the chickpea or our standard soybean. And then the last one is our safflower relay treatment. Here we planted the camelina on 15-inch row spacing, and then the safflower is also seeded on 15-inch row spacing. So alternative rows for that safflower crop. And the rationale for this is really just trying to explore what other crops might work within this relay cropping system to ideally be able to expand the acreage of winter camelina that we can get out on the landscape. So this study was done starting in fall of 22 through summer of 25. We did 2 growing seasons with each crop. This graph is showing our monthly precipitation. So the long-term average are the blue blocks, the light blue blocks in the background, and then the dark blue bars are showing our precipitation for each of the years. So I just wanted to draw attention to some of the precipitation issues that we had. So our first spring, April, May, June of 2023, was very dry. It was much drier than normal. All 3 of the crops were grown in this year, so we had all 3 present and we had this dry season. The following 2 years, in 2024 and in 2025, we had much wetter springs. And so 24, we had chickpea and safflower present. And that was because we had a lot of drown out in our camelina. And so we had to kind of modify the study to get enough replication. So we had to cut out the sunflower treatment, and we just had the chickpea and safflower present that year. And 2025, somewhat of the same situation because it was so wet in the spring. We had some loss from our camelina, but we did have sunflower present. I don't know if I said that wrong. 24 was chickpea and safflower, 25 was sunflower. So really dry the first year and really wet the second year for all three of the relay crops that we trialed. So I'll start with our chickpea data. So we've got, this is NDVI for chickpea in the 22-23 and then 23-24 growing season. And I'm just showing this starting in the spring of the year through the fall. The purple line on both graphs indicates when the camelina was harvested. So our chickpea would have been seeded in May, and then the camelina harvested in early July. On the top, you can see a comparison of the monocrop versus the relay crop chickpea the day after we harvested. So in 22-23, you can see that monocrop chickpea has a really good canopy development to it. You can also see that in the NDVI, so that light green or light gray bar after harvest, much higher canopy cover, whereas our relay crop was much more stunted. In large part, this is because of how dry it was this year and the competition for moisture. So we had that much lower canopy cover. And then as we moved into August, we saw that canopy really fill out for the chickpea. In the 23-24 year, much more similar between the two treatments. And this is primarily because the canopy of the monocrop was set back, and then the canopy of the relay crop was a little bit better because it had some of that extra moisture. So on the NDVI graph, you can see those two canopies looked much more similar following camelina harvest through the end of the season. For all of these systems, we looked at population, height, growth stage, yield, for chickpea protein yield or protein content, and then protein yield, and compared that monocrop versus the relay crop treatment. In the first year with chickpea, we saw a much higher yield in our monocrop than our relay crop system. So significant reduction in both seed yield and protein yield for the relay crop. But in the 23-24, the only significant difference between the two treatments was the height at the time of chickpea harvest, but nothing in terms of yield or protein content or protein quality. But if you look at those yield values for both treatments, we're right around 15,000 kilograms per acre. In 22-23, both treatments had a higher yield than that. So the chickpeas seemed to really prefer that drier growing season, which allowed them to do much better. So we didn't see the yield difference the second year, but chickpea overall performed less well under those really wet growing conditions. Sunflower, very similar story. So again, in the dry year, 22-23, we see this big difference in our monocrop and relay crop canopy development. In 24-25, where we had that wetter spring, that difference, the monocrop canopy was still a bit higher initially, but by the end of July, the two crops had really caught up similarly. So that dry year, we see a much bigger difference in their development early on compared to that wet year. In terms of the actual crop performance, that first year, the relay crop was basically abysmal. So we had about half the stand. The crop was very short, as you can see, very etiolated, much lower yield, our oil content, and then our oil yield was all lower. So the sunflower that year, because of how dry it was at planting, it really had a hard time establishing and then a hard time competing for that moisture, and really just got set back. In the second year, similar to the chickpea where we had more moisture, we did not see that same effect. So no significant difference across any of the response variables. So you can see in the picture after harvest, that canopy looks a lot more similar to our monocrop. The one thing with the safflower or the sunflower that we really had to watch is in the second year, that crop was much taller, so it was more similar to the monocrop, and we had to be careful with our harvest combine height. So with the sunflower, if we cut those heads off, we basically lose that production yield from a plant. So in a better growing year for the sunflower, there's more complications with harvesting the camelina. And then finally, our safflower, again, is very similar story. You can see the safflower plots in that dry year just really looked pretty rough. The monocrop looked good, but in the relay crop really did poorly, and they never really recovered. So if you look at that NDVI, it just always stayed pretty low, and that's mostly due to the patchiness of the stands. 23-24, a little bit of a delay in that relay crop, but once they were able to grow out of it, a much more competitive crop relative to the monocrop. And again, interestingly, we did not see a yield difference in the 23-24 growing season, but some of that had to do with the variation across the plots, and yeah, some of the removal of certain areas where we just had poor stands of both camelina and safflower. So I think this might be a bit misleading, especially where you look at our relay crop population was much, much lower than the monocrop population. So this might be a little bit of a false narrative with this yield here. However, in 23-24, we also didn't see a yield difference similar to what we saw with the other plots. So across all of the variables outside of the height, we saw a much more consistent performance between the monocrop and the relay crop. So the story of it is really, one, the environment is having a huge impact on this. So our chickpea liked the drier year, our safflower and sunflower liked the wetter year. I would say overall, just observing the crops, chickpea was much better adapted to the relay crop system based on its growth habit, how it responded to being in the canopy, ease of harvest due to it being multi-branching. And if you clip some branches, you don't have to worry about a major yield loss because it's able to send out a lot more branches. So chickpea looks like it has some real potential to be relay cropped. We need to do more work looking at different varieties. We grew CDC Palmer. There's a number of other varieties that could be tried. So I think this could have a lot of potential, especially as we start to shift a little bit west where growing conditions are more consistently dry than here in Morris. The sunflower and safflower, we did see more establishment issues with those crops, and they just overall, looking at them in the field, had some more issues. We know sunflower we can do in a double crop system, so that's probably going to be the preferred method with these winter oil seeds. And safflower was just not a great crop for this region. So all have some potential, but would definitely need more agronomic work to move forward with them.
Carrie, did you say that the safflower did better in the wetter year?
It did because it established better. But I think the yield was worse. So no, it did not. There was better establishment. There were better plant stands. The plots looked better, but we had a lot of aborted seed. So we had a lot of empty seed that year. That was really wet, and it was also pretty cool for most of the growing season. So the plants in general looked better. The yield was pretty terrible.
What variety of camelina did you use?
This was with Joel.
Joel, okay.
Yeah.
Yeah. If there was a fast way to harvest safflower as a spice, that would be better, but that's hard to do.
Yeah. Well, we can look into that because it didn't set seed great in that second year, and the seed is a pain to clean. So maybe the spice is easier.
Okay, so I'm going to try to breeze through this really fast. Part of it is on herbicides, which I know very little about, although I did see Mike Bernard's on this call, so if anybody has any questions, I can ask him. But I'm just going to report on a few results from a couple different studies that we began last year. One of these is looking at the volunteer persistence of pennycress, basically yellow versus black. And we know that the yellow pennycress, part of the reason for that yellow coat is because the fiber has been reduced in this. This, I think, is a TT8 mutant, if I remember correctly. But anyway, it's a thinner seed coat, helps give it that a yellow golden color, and it's less dormant or no dormancy at all. And indeed, observationally, anecdotally, that we have seen this out in the field where we've had yellow and black pennycress grown side by side. We tend to see come back the next year after harvesting the pennycress, there are far fewer yellow pennycress volunteers than there are black. In this study here, soybean was grown right after the pennycress harvest, so a common soybean herbicide would have been used here, and it apparently knocked back the yellows, but there's still quite a few black pennycress that volunteered in the next year. So with this study, we wanted to document that experimentally, and we'll be publishing this too, of course. But is of course to compare the volunteer persistence between yellow and black pennycress, and this is in a spring wheat, pennycress, double crop soybean, corn rotation. So Mark Bernard started this. We no longer have Mark Bernard, so I inherited this study. The pennycress was planted in 24. The fall was very, very dry. And so most of that pennycress really didn't emerge until spring. So it was kind of a late harvest for the pennycress, July 9th. We harvested that. We burned the whole area down with paraquat to get rid of any weeds or any potential volunteers that were out there at that time, the pennycress. And then immediately or the next day planted soybean. And that's where we applied our treatments for this study. Mark had 5 treatments. I reduced it to 3 because I just had too many other things going on. But the treatments here to use common soybean herbicides that would be used with soybean to see what effect they have on this volunteerism of pennycress. So the 3 treatments, we had a pre-herbicide with, I think it's a long residual, Mark can verify this, but a relatively long residual, applied after soybean planting, Tendovo, which is metachlor, metribuzin, and however you pronounce this one. And then a second treatment, which was to make a post-herbicide application with short residual, applied about 3.5 weeks after soybean planting. And that was composed of 2,4-D and glyphosate. And then the third treatment is both the pre and the post. So we measured pennycress emergence before adding the post application, a few days before adding the post application, 7 weeks after the post application on September 25th. And then we measured again before the bottom dropped out on the thermometer and the soil froze up, which was November 19th for us. And we will be following that again this spring to see before planting the corn. We'll see if there are any volunteers out there in the black and yellow pennycress plots. So this kind of summarizes the data that we collected from this first year. We got yellow pennycress here on the left, black pennycress. Density, and this is plant density, of course. As you can see here, the August 1st density counts, both are quite high. This is before making the post application, okay, in the post treatment here. So that is pretty much what came up after harvesting the pennycress. But you can see, even though there's a lot of volunteers, there happened to be more black than there was yellow. The yield of these two, the black and the yellow pennycress, was not that much different. It was not significantly different. So apparently there is maybe a little more shattering of the black than there was of the yellow. But you can see at this time the pre is already starting to take effect. Had a bigger impact on the yellow pennycress, as you can see from the y-axis. The numbers are quite a bit lower than they are, the red bar here, for the black pennycress on the first counts. But then by September 25th, and of course this is 7 weeks after that post application, the pre's had time to work. It's done a real good job on the yellow pennycress. The pre plus post treatment totally annihilated the pennycress. And the post treatment did very well too for the yellow pennycress. There's only, a few plants out here and there, but not too bad. But there's still plants there for the black pennycress, even after the post application, and even by November 19th, when the final counts were made, there's still a lot less, but there's still relatively, what I would say, be too many plants out there after these treatments. So the results so far indicate that most of the volunteer pennycress does germinate within a few weeks after harvest, relatively short time, and it looks like it can be easily controlled with common soybean herbicides. So this is some data that I'm going to report on. This Greg Johnson and Mark Bernards have been working on, and this is looking at identifying herbicides after short season summer crops, used with short season summer crops, that may or may not impact the growth, emergence growth and yield of pennycress and winter camelina. Greg couldn't be here. He's on a NEFA review panel, grant review panel, so he couldn't be here. And I think Mark is on this call, or at least he was. I don't know if he is now. But anyway, I'm primarily going to report on the results from the Waseca site, because Greg gave me those results, and a little bit on Morris too, and just for the first year of this study, which is funded by the Forever Green Initiative. So one of the things Greg wanted me to point out is that pennycress, generally speaking, winter camelina does a little bit better down in Waseca than pennycress, and sometimes they get poor pennycress emergence. In the plots that were used for dry bean and peas, for these legume crops, which are short season summer crops, pennycress did not emerge very well, so you're only going to see results for camelina here. Also, he didn't have any error bars on this, so I don't know about the variability. But the bottom line here with dry bean is that the use of dual magnum, which I believe is metallochlor is the main ingredient there, had little effect on camelina. It had little effect. I believe Mark did this in Morris too. It had little effect on either camelina or pennycress at the Morris site. But when you add these post treatments like Reflex and Raptor and also Permit, we start to see there appears to be some negative effect there. A little bit different, well, not too much different at Morris. These are some results that Mark gave me here from the Morris site. You can see metallochlor doesn't appear to have, as a appears to have no effect on camelina emergence and yield. But when adding the Reflex as a post, you can see there was a lot of damage. This is at Morris, a lot of damage. And that kind of verifies what Greg saw too down there in Waseca. Raptor, however, apparently not as much damage, whereas at the Waseca site, there was lower yields. And these other ones, Permit, that was one that apparently had some effect at Waseca, and you can see here in Morris, there was quite a bit of damage to the camelina as well for that one. Peas, I don't think we did peas at Morris, but in Waseca, again, poor pennycress emergence. So Greg just dropped pennycress out from this. But camelina, maybe a little effect on stands there. But as far as yield, the only perhaps optil as a pre seems like it may have had some effect on camelina yield. But with these others, dual magnum and the post treatment, no effect there. Wheat, we've seen in the past typically that the wheat herbicide or herbicides that are commonly used on wheat don't affect pennycress and camelina all that much. Certainly pennycress, both stands and yields were little affected at Waseca by these different common wheat herbicides. I should point out, I forgot to on the last slide, the furthest to the left here is the weed-free control that's being compared to here. Okay, forgot to point that out. Hopefully you probably assume that anyway. But anyway, yeah, little effect on pennycress, but there were some winter camelina yields that were low on some of these herbicide treatments. However, when I talked to Mark about this, he said in Morris there was little effect of the same wheat herbicides on camelina at the Morris site, and that's shown here in these results of injury caused by the application of those herbicides prior to planting camelina and pennycress too. Here's the untreated weed-free check, and you can see very little damage as compared to the weed-free check. Sweet corn, of course, both wheat and sweet corn are monocrop. They're monocots. Pennycress and camelina are dicots. So generally speaking, monocot crops like that, herbicides that are used on them, generally don't affect the dicots as much. Of course, Mark may have different thoughts on that. But sweet corn, like herbicides for sweet corn, like wheat, had little effect on either camelina or pennycress. However, I did notice that camelina or pennycress emergence and yields were fairly low at Waseca, so probably need to be a little bit comparing these results. But generally speaking, it looks like the sweet corn herbicides had a little effect on either camelina or pennycress. So the bottom line is, obviously, herbicide choice is going to be a very important consideration when planning to plant pennycress or winter camelina after these short season crops, because for the most part, it seems like several of them do not have a deleterious effect, but some do, and so we have to be conscious of that. Greg wanted me to point out the work is ongoing. There's still more data analysis that needs to be done and data collection as well before making any recommendations. The Waseca site Greg will be collecting data again this year. The Morris site, we had to cut that site. We just don't have the resources to do it at Morris.
For the black seeded pennycress compared to the yellow seeded, were you kind of suggesting that, it's not like volunteers are not even a problem for black seeded because you can control them with a herbicide following?
That's what we're hoping for. But as you can see,
yeah.
Oh, go ahead.
I was going to say that it would be a problem if you want to grow pennycress there again, though.
Right.
Because then if you're getting different varieties, you're going to get them all mixed up.
Yeah. Yep. So we've gotten to a total different area for the repeat. Are you talking about the repeat of this?
Yeah.
Yes. But you can see here, even by November 19th, and this is before the soil froze up, there's still plants there for the black seeded. My guess is, I haven't looked at it yet. Haven't been that far down into the Swan Lake yet, although most of the snow is melted now. But I'm guessing there's probably still going to be some plants here in the black seeded. But the main thing is the yellows. It looks like they're knocked out pretty good by those soybean herbicides. Wouldn't you say, Mark?
And remember, those scales are different.
Keep in mind different in scales.
Yeah.
And the real goal of this study was to demonstrate that you wouldn't have carryover of volunteer pennycress past that first summer to hence people's willingness to adopt it as a crop, because most people, when they hear pennycress, say, why do I want to plant a weed in my field? And we need to be able to demonstrate that with effective weed management and soybeans, you can eliminate it with the goal.
But you're only
It's not
seeing
gonna happen
1
with the
year of
black.
data though, like, this didn't carry over to the next year to see like how much more black seed comes up the next year.
Correct. That data will be collected.
We are doing that.
Okay.
So we're remember, corn is in the rotation too. So we're going to be looking at it. It got repeated this past fall. So we have another cycle, and it's in a different area, not this same area, so that we don't have confounding there. But we will look at the emergence before we plant the corn. And actually, after we harvest the corn, we'll still go back to those plots to see if there's a, because the corn, we'll use a normal herbicide regime for the corn too, which may or may not, if there's any volunteers, may or may not affect them. Of course, if we hit it